Okay, let's come to the final part of our lecture, which is the anatomy of the iris. So, what is the iris? The iris is the most anterior part of the uveal tract, act as a circular diaphragm in front of the lens and the ciliary body, separating the anterior chamber from the posterior chamber. And its color is the color of the eye, meaning that it will depend or it will determine the color of the eye. So this color can be different according to the race, and it is due to melanin. So increasing the concentration of melanin will lead to darker color of the iris, while decreasing the melanin will cause whiter or more lighter color of the iris. 12 millimeters in diameter, with the thickness more at the collarette and the thinnest at the root. So always try to remember where is the thickest location and where is the thinnest location. It's very frequently asked in the exam. So the thickness is 0.5 millimeter at the collarette, while it is thinnest at the root. Here is an anterior view of the iris. So if we, this is the anterior surface of the iris, and this is will lead to this is which is responsible for the color of the iris. You may be surprised to know that the posterior surface of the iris is black, despite the race and despite the color of the anterior surface. Them frequently. So they ask if what is the color of the posterior surface of the iris, and whether it will be different according to the melanin content, and the answer will be no. It will be the black, even with a very light color iris. So if we have a section or we have a view of the posterior surface of the iris, what can we see? So here is the pupil, and we have two types of folds. We have, so as I told you, the color will be black, and we have vertical folds extending from the pupil to the ciliary body, or to the ciliary margin, which is called the radial furrows or Schwab's contraction folds, the vertical one. While the horizontal one, this is called radial contraction folds or circular furrows, which is more prominent at the iris root. So we have radial furrows and we have circular furrows. So what about the anterior surface of the iris? We have the some important landmarks, which is called the iris crypts. So the iris crypts are depressions within the surface of the iris, and we have the collarette, which is the thickest part of the iris, which acts as a ridge encircling the pupil and separated from the pupil by about 1.5 millimeters. And this collarette will separate the iris, or the anterior surface of the iris, into two zones. The inner one, which is called the pupillary zone, and the outer one, which is called the ciliary zone. So this is the inner one, the pupillary zone, and the outer one, which is the ciliary zone. Here is the pupillary zone, and here is the ciliary zone, and separated by the collarette. So the collarette is like a zigzag-like structure, a ridge about 1.5 millimeter from the pupillary margin, which it marks the site of circulus iridis minor. So at the end of lecture, we will say that the circulus iridis major will give supply to the circulus iridis minor, and since the circulus iridis major is present in the ciliary body, the circulus iridis minor will be present into the collarette. The embryological importance of the collarette, that this was the site of attachment of the pupillary membrane. So the pupillary membrane was a membrane that was closing the pupil during the embryonic life, that disappeared at the 8th month of gestation, and it was adherent to the collarette. Sometimes we can see some remnants of this pupillary membrane clinically in some patients, and within the collarette, there are some like depressions. These depressions are called the Fuchs iris crypts. So they are pit-like depressions in the region of the circulus iridis minor. So in the newborn, there is no collarette or crypts, but they develop later on in life. To differentiate between the pupillary zone and the ciliary zone, so the pupillary zone is relatively flat, Well, sometimes the pigment frill will be extending outside or running over the pupillary margin, while the ciliary process or the ciliary zone will contain the radial ridges again, which is formed by passage of blood vessels from the circulus iridis major toward the circulus iridis minor. And also it has contraction furrows. So here are the contraction furrows. It will deepen with pupillary dilatation. So again, here are the radial folds, radial folds, radial folds present in the ciliary zone, and here are the these radial folds are due to presence of the blood vessels running from the circulus iridis major in the ciliary body to the circulus iridis minor present in the pupillary zone at the site of collarette, while this is the circular contraction furrows. These are the contraction furrows which will deepen on the pupillary dilatation. The color of the iris will depend mainly on the amount of melanin in the anterior border layer. So if we have a slight amount of melanin, this will lead to blue color of the iris. If we have moderate amount of melanin, this will lead to hazel color of the iris. Large amount of melanin will lead to more darkish color, which is brown color. So let's now talk about the structure of the iris. So the iris is formed from stroma, from epithelium, and finally from smooth muscles. So stroma, epithelium, and the smooth muscles. If we have here a section of the iris, so let's see what are the arrangement of the iris layer from anterior to posterior. So the first one is called the anterior border layer here. It is like, again, a condensation of the anterior stroma, very thin layer, followed by the stroma of the iris. Here is the stroma of the iris. And the stroma of the iris containing the iris sphincter muscle, which is a muscle present near the pupillary margin to constrict the pupil. This is followed by the anterior epithelial layer, and the anterior epithelial layer is continuous with the dilator pupillae muscle. So the dilator pupillae muscle is present near the ciliary margin, and it is a myoepithelial cells or myoepithelial muscle. So these basically are epithelial cells with a muscle element. And then we have the posterior pigment epithelium of the iris. So the posterior pigment of the iris is the most posterior structure, and this is what makes the iris looks black from behind. This is other structure, this is the reflection of the posterior margin of the posterior epithelium, which is called the pupillary ruff. So this doesn't occur in all individuals, but it can occur normally. It's forming what's called ectropion UV, as you can see, I think here. Here is the ectropion UV, which is just extension of the posterior pigment epithelium of the iris forward. Okay, so the anterior border layer, as I told you, it is a condensation of connective tissue and pigment and the pigmented cells from the condensation from the anterior stroma, developed at the 7th month of gestation. Developed as the 7 months of fetal life, and then undergoes generalized atrophy at the pupillary zone, forming this iris crypts, and irregular atrophy at the ciliary zone. So it will be generalized atrophy in the pupillary zone and irregular atrophy in the ciliary zone, forming this iris crypts. The anterior border layer is absent at the pupillary zone and only present in the ciliary zone. This is important to be asked in the MCQs. So the anterior border layer is not present in the pupillary zone, only present in the ciliary zone. And this one which contains melanin, so this is the one which is responsible for the color of the iris. Then the thickness of the stroma is formed, the thickness of the iris after the anterior border layer is formed by the stroma, which consists of, like any stroma, which consists of collagen fibers, consists of blood vessels and the nerves and the melanocytes. within the glycosaminoglycan matrix, and has more elastic tissues and fewer pigment cells than the anterior. And one of the also asked questions in the exam that it has a similar appearance irrespective of the color of the iris. So whether the color of the iris is black or light, the stroma will be the same. Then after the stroma, we have the pigment epithelium, and the pigment epithelium will be either anterior or posterior. So the anterior pigment epithelium, we said that in the embryology, we will say that it will derive from the outer layer of the optic vesicle, while the posterior epithelium will be derived from the inner layer of the optic vesicle. The anterior epithelium layer, which is the forming of the dilator pupillary muscle. So as you can see here, the epithelial layer has a superficial or the apical part of the cell is transformed into a myoepithelial portion. Sorry, the basal part is transformed into myoepithelial portion. So these are the basal part of this basal part of the cells will act as a muscle, which is the dilator pupillary muscle. While the posterior epithelium is covered with basement membrane that is continuous with that of retina and the ciliary body, with high amount of melanin and basal nucleus and basal enfoldings, with lateral walls are joined by desmosomes. So this is important in asking the exam, where is the myoepithelial portion? It is into the anterior epithelium. Where is the basal enfolding and the high melanin content? This will be in the posterior epithelium. And sometimes the posterior epithelium will reflect forming this frill or pigment frill or ectropion UV. And the lateral walls of the posterior pigment epithelium are joined together with the desmosomes. So here is the anterior layer. The basal part will show this is the dilator pupillary muscle, while the cells are joined apex to apex. And the posterior pigment epithelial layer will show high melanin content. This is why it will give the posterior surface the black color. We have two muscles of the iris, mainly the sphincter pupillae and the dilator pupillae muscle. So this is the function of all of them. This is the normal pupil in the normal light. With exposure to light, this will lead to a stimulation of the constrictor pupillary muscle by the parasympathetic pathway, while with dim light, the pupil will need to increase its size to increase the amount of light entering the eye, and this will be the effect of contraction of the dilator pupillary muscle, which will be sympathetically innervated. So constriction of the pupil is under the parasympathetic, while dilatation of the pupil is under the sympathetic. So parasympathetic will cause constriction of the pupil by the constrictor pupillary muscle, which is a circular muscle arranged around the pupil. While that's why when it constricts, it will lead to decreasing the size of the sphincter. While the dilator pupillary muscle is present near the ciliary zone, that's why, and act as radial. So this radial fibers, when contract under the sympathetic innervation, will lead to dilatation of the pupil. The blood supply of the iris comes from the circulus iridis major as well, with anastomosis of the two long posterior ciliary arteries and 7 anterior ciliary arteries. So who will say to me where is the missing anterior ciliary artery of which lateral rectus? So I said the answer is lateral rectus. So we have two long ciliary arteries for each rectus muscle, except for the lateral one. And it's present into the ciliary body. So you have to know that the circulus iridis major, it is only arterial, so there is no venous major iridis circle. While the minor iridis circles or circulus iridis minor, which is present at the site of collarette, it is receiving radial branches from the circulus iridis major present into the ciliary body, and it has both arterials and venous. So it is arterial and venous, the circulus iridis major. This is again asked frequently in the exams. So here is the circulus iridis major present into the ciliary body, passing radial branches to the circulus iridis minor present at the collarette, which has arterial and venous. The venous drainage of the iris again from vortex veins, it is the superior and inferior ophthalmic vein to the cavernous sinus. And there's just a hint about the capillary endothelium of the iris. It is non-fenestrated with tight junction that prevent the passage of large molecule, and this is what comes to what's called the aqueous barrier. We will not go through this. It is we'll discuss this thoroughly in the physiology. So just to know that the blood aqueous barrier is formed by the vascular endothelium of the iris, which is non-fenestrated with zonular occludens and the tight junction of the ciliary epithelium. So this is the tight junction of the ciliary epithelium or the zonular occludens and the non-fenestrated capillaries of the iris. This will form what's called blood aqueous barrier, which has a function to prevent certain molecules to enter within the anterior chamber, maintaining its clearance like plasma proteins. We have different barriers in the eye, such as blood aqueous barrier, blood retinal barrier, and this will be discussed in the physiology. So the nerve supply of the eye will be through the parasympathetic, or the nerve supply to the muscles of the iris will be through the parasympathetic, through the third oculomotor nerve to the will relay in the ciliary ganglion, and it passes through the short ciliary nerves to supply this the constrictor pupillae muscle and the ciliary muscle, causing pupillary constriction, while the sympathetic pathway will pass through the first order neuron of the hypothalamus, second order neuron of ciliospinal center of Budge, third order neuron of the superior cervical ganglion, then the postganglionic will pass with the nasociliary nerve to the long ciliary nerve to the iris dilator muscle, causing dilatation of the pupil. We have another sympathetic supply to the iris vessels, which is to the wall of the blood vessels, again coming from the sympathetic root to the short ciliary nerves. So the sympathetic nerve supply to the iris, either through the short ciliary nerves to the wall of the blood vessels, or through the long ciliary nerve to the dilator pupillae muscle.